Well, she's a very, very well-known scientific. She's the head of the Laboratory of Molecular Endocrinology in Valencia and the Centro de Investigación Príncipe Felipe, where she is also the director of the center. In Valencia, are very lucky with the direction of Deborah, of course. She did her PhD studies in the Vanderbilt University, and then he had several postdoctoral trainings in very important universities in the United States, in Duke University, and also in the Harvard Medical School. Well, she comes to Spain several years ago, and she has done a very interesting career here in Spain, dedicated to several research lines, especially signal transduction pathways in human health, especially related with the role of insulin receptor signal transduction in the regulation in a lot of diseases such as aging, obesity, diabetes, and neurodegeneration. She has a very interesting curriculum with more than 60 peer-reviewed papers in very interesting journals such Nature and Nature Genetics. So for us, it's a pleasure to host you, Deborah. Thank you very much for coming.
To you and Jordi for organizing this initiative. Also, I'd like to say hello to Itty Bobs, to my colleagues there. I have several collaborators from Fieber de Vim who are Itty Bobs. And thank you all for being here. I realize it's Puente or almost Puente in several areas of Spain, which makes your attendance today even more appreciated. Thank you very much. What I'm going to talk about today are several themes related with what we do in my laboratory, which is to define the molecular basis of insulin resistance and its implications in diabetes, obesity, and chronic liver disease. We have additional interest in other aspects of insulin signaling, for example, in the central nervous system, in terms of appetite regulation, as well as modulation of cognition. But today, because it's a liver seminar, obviously we're going to focus on insulin resistance. In the first slide, I'd like to take the opportunity to remind you that as the prevalence of obesity has increased, so has the prevalence of these forms of hepatic disease. And also to comment what my perception of the term liver plasticity is. When one of my postdocs saw the title, they said, well, what do you mean by liver plasticity? We're not talking about the central nervous system. And for me, plasticity is completely appropriate for the liver, given that it has a huge regenerative capacity. And many of the pathways, both cellular and molecular, that are involved in these distinct at each of these distinct phases, there are molecular defects underlying the pathology. And in my perspective, these molecular mechanisms have a certain amount of plasticity. So the pathological process can go forwards, and until a certain point, and I think clinical as well as basic research studies demonstrate this, that the process may be reversible. So that's simply what I mean about plasticity. That whenever we talk about, thank you, that whenever we talk about plasticity, it's that some change could be made to the process. I also have to declare in the beginning that I'm from one of these orange states, from Alabama, where the incidence, and I think this graph, this map, clearly reflects the extent of diabetes, obesity, and chronic liver disease for the healthcare system. Because in states like my own, Alabama, the incidence, and these are statistics from several years ago, and the actual statistics show that around 30 to 35% of the population has type 2 diabetes or is in a pre-diabetic state. We see one of the basic reasons for this, which is the high prevalence of obesity in these states. In Alabama, almost 40% of the population display excess body weight or obesity. And these maps reflect perfectly the relationship between obesity and diabetes. And these diseases share some of the features with chronic liver diseases, with NAFL, fatty liver disease, and the more advanced form of that, of NASH. As most of you know, type 2 diabetes is a multi-system disorder. For many years, it's been hypothesized that peripheral insulin resistance is the major molecular cause of type 2 diabetes. During the last 10 or 15 years, we've clearly recognized through both clinical as well as studies in preclinical models that beta cell failure, so insufficient beta cell function or mass, is also a critical factor in the development and progression of type 2 diabetes. And at the end of the talk, I'd like to come back a little bit to this parameter that I think is also not a trivial factor in NAFL and NASH, particularly in the progression of these disease states to cirrhosis or perhaps even hepatic carcinoma. One of the principal objectives of our laboratory is to understand how these defects in insulin sensitivity contribute to hepatic dysfunction. One of my brothers is a physician, and he commented to me recently related to the maps and the statistics I've just presented, that the difference these days between the service, the clinical service, the clinical department of endocrinology, hepatology, and family medicine are clearly not very distinct anymore, because many of the physicians in each of these departments are seeing the same profile of patients, which are generally obese patients with either metabolic syndrome or diabetes, who present with some sort of hepatic dysfunction. We know through the research from the last years that one of the underlying pathological factors is related with the role of excess adipose tissue accumulation as a principal source of inflammatory cytokines. For many years, at least in the field of diabetes, we've recognized that insulin resistance is either the cause or the consequence of chronic inflammation. This chicken and egg argument, I think, still needs to be resolved within the field, but the clinical reality is that obviously excess accumulation of adipose tissue generates excess inflammation, and also, of course, in insulin-resistant states where insulin is no longer able to properly regulate lipid storage and lipolysis, there's a liberation, a constant source of free fatty acids circulating in the body. My laboratory focuses, this is what I've done for most of my career, some sort of work in signal transduction. And my laboratory focuses in different areas of investigation, but always with the same principal question, which is how insulin signaling regulates basic cellular processes such as proliferation, differentiation, and apoptosis. And we're very interested in applying these basic questions to understanding the basic mechanisms of metabolic diseases. Once insulin binds, or the related hormone IGF-1 bind to their cell surface receptors, both receptors for insulin and IGF-1 contain intrinsic tyrosine kinase activity, and the binding of these hormones activates the intrinsic tyrosine kinase activity In turn, these activated receptors are able to phosphorylate IRS proteins, insulin receptor substrate proteins, which are the principal intracellular substrates for these receptors. IRS molecules are very large and contain a number of different sites for both tyrosine as well as serine threonine phosphorylation. The phosphorylation by the insulin receptor converts the sites on the IRS proteins as binding sites for other components of intracellular signaling, such as the PI3 kinase AKT pathway. And once these pathways are activated, IRS proteins orchestrate the cellular response to these hormones to regulate metabolic events such as the incorporation and utilization of glucose. So these IRS proteins are able to orchestrate, to regulate these intracellular events that lead to insulin-regulated gene expression, activation of proliferation pathways, regulation of the cell cycle, or in the case that we're going to discuss today, regulation of carbohydrate metabolism pathways or lipogenic pathways in the liver. Many years ago, in order to test the function of different IRS molecules, we generated different knockout models. So knockouts of IRS1, IRS2, IRS4. And we observed that IRS2 has a specific function in carbohydrate as well as lipid metabolism. IRS2 deficient animals develop a rapid and severe form of diabetes due to the role of IRS2 in both pancreatic beta cells as well as peripheral insulin-sensitive tissues like muscle, fat, and liver. And what we study with this model of IRS2 deficiency is the interaction between these various points of defects that are related to the development and progression of type 2 diabetes. We're particularly interested in the axis between the liver and the pancreas, as well as between the adipose tissue, the pancreas, and the liver. And these are some of the new studies that I'm going to present today, which broaden a bit our perception of IRS proteins and how they may regulate crosstalk with other signaling pathways, such as the FGF pathway. We know, as I mentioned, from the studies of the IRS2 knockout, that these animals display profound insulin resistance at the level of the liver. In consequence of this, they develop a phenotype in the liver that's similar to fatty liver. We encounter deposits of lipids in various areas of the hepatic tissue. These animals also have a defect, obviously, in glycogen storage. And some of the studies from other laboratories, such as Goldstein and Brown, have demonstrated quite clearly that IRS2 is a fundamental molecule in regulating lipid metabolism in the liver. During the last 10 years, there's been an ongoing controversy about what is the specific role of IRS1 versus IRS2 in the context of hepatic function and metabolism. This schematic that I've presented here, in my opinion, is a bit oversimplified because it attributes the functions of IRS1 to carbohydrate metabolism, to the regulation of genes involved in the gluconeogenic pathways. Obviously, if these animals are insulin resistant, they have alterations. They have very defective glucose production in the liver. This is one of the reasons why they display hyperglycemia at an early age and die from diabetic complications, is the chronic state of hyperglycemia. IRS2, in contrast, is attributed in this schematic, and I think there are data to support these conclusions, may be more involved in regulating lipolytic genes and the function of lipid metabolism within the liver, particularly in response to insulin and the crosstalk between these pathways. These are papers during the last few years that have tried to clarify the role, the distinction between what is the function of IRS1 and IRS2 in the liver context. Obviously, if these two knockouts provide two different phenotypes in mice, one is a diabetic normal size mouse, the IRS1 knockout is a small growth deficient mouse model. It gives a context for predicting that they do have different physiological roles, particularly within the liver. The laboratory where I did my postdoc, Morris White, as well as other colleagues from that same era, have generated all sorts of different transgenic knock-in, knockout, liver-specific models to try to tease apart and elucidate a clear model of what the functions of these IRS proteins in liver is. Some of the data that I'm going to present today, I think, serve to clarify the role of IRS2 and clearly how it's related to regulation of hepatocyte differentiation, as well as accumulation of lipids within the liver. This is a study that was published years ago that provides another piece of evidence to demonstrate the importance of IRS2 in the context of liver function. This study, and there are other studies that confirm these observations, that a large percentage of hepatic carcinomas display an overexpression of IRS2. This data will become a little bit relevant in the final part of the talk related with the crosstalk between IRS2 and FGF in the progression to a more severe state of dysfunction. This is a paper that was published this year. It's a paper that we would have liked to publish. Our paper is under revision at the moment as some of the studies that were published in this work from Dr. Lim's group in Korea address some of the basic questions that we have about IRS2 as well. And one of the basic findings in this paper is that deletion or loss of function of IRS2 prevented the development and progression of tumor formation in livers that were susceptible to fatty liver disease. This confirms some of the previous work linking IRS2 to progression of the liver disease spectrum. Based on these observations and these unresolved questions about IRS2, also related with other projects that we have in the laboratory, which examine how insulin and IGF-1 regulate stem cell function, we've applied the methodology and some of the tools we have in the laboratory to investigate how insulin regulates the behavior of liver progenitor cells and what the role of insulin resistance may be in liver repair, basic liver repair mechanisms. One of the reasons to address this question is actually because there have been very few studies which have addressed the role of insulin and IGF-1 on stem cell niche behavior, on migration and differentiation of progenitor cells within the context of normal physiology. As you know, insulin or sensitivity to insulin is a very important factor in defining quality of life as well as longevity. Many models from C. elegans to preclinical models such as genetically modified mice have shown that manipulation of the insulin pathway has a drastic effect on lifespan, as well as different components of aging-associated diseases. Stem cells represent an endogenous natural repair mechanism for responding both to tissue damage as well as the natural process of aging in tissues of the human body. So the two fundamental questions that the presentation today address are what is the role of IRS2 in the activation and differentiation of liver progenitor cells, and how insulin resistance may regulate, may disable these biological, physiological processes of repair within the context of the liver. As I mentioned before, we have a pretty clear idea of how these IRS proteins are regulating basic metabolism, glucose uptake, lipid storage, but it's still unclear. I've done it again. It's still unclear how IRS signals may regulate basic aspects of cell proliferation and differentiation. In order to address these questions, we had to develop a series of tools. And this is just to remind you that, and I know it's controversial in the liver. Actually, my background is much more in another endoderm-derived tissue, which is pancreas. And we continue with the controversy of, do progenitor cells actually exist in the pancreas? And if so, what is their niche? What is their role in actual liver biology? These are some of the same questions that we have in pancreas. Liver progenitor cells are bipotent and are capable of differentiating under appropriate stimuli to either parenchymal hepatocytes or alternatively to cholangiocytes. In order to study how IRS2 insulin promoted signals regulate this process, we had to establish some basic experimental models in the lab, because ultimately we wanted to apply this knowledge of IRS regulated progenitor differentiation to understanding more about the progression or reversion of the different states in the liver disease spectrum. And the next slide is a basic model of the activation of the different molecular cellular processes that characterize, that underlie fibrosis in the liver. And one of the fundamental questions that we've tried to resolve is how insulin resistance in the particular studies that you're going to see, characterized by the loss of IRS2 function, might alter or accelerate the process of fibrosis in the liver. In the next slide are some observations from a published study which demonstrate that one method for regenerating or studying the process of liver damage, partial hepatectomy, triggers the accumulation of IRS2 positive cells in parts of the liver that we would associate with the triggering of a regenerative response. This is a comparison between normal and IRS2 mice, which obviously, if they're deficient in IRS2, don't have an accumulation of these cells. And one of the questions that we're going to get to in just a moment is, what is the role of IRS2 in response to models of hepatic damage? In the next slide is a summary of the IRS2 deficient model. Because we believe this is an excellent model for actually teasing apart, for dissecting the molecular cellular basis of pathogenesis, such as fibrosis related with NASH and more advanced stages. As I mentioned, this mouse model is profoundly insulin resistant, particularly at the level of the liver, due to a defect in the beta cell function. These mice are also hyperinsulinemic. I mentioned before the fundamental role of IRS2 in regulating the development and maintenance of the pancreatic beta cell mass. These animals, as you can also see from this photograph, also develop mild obesity. But the model displays a sexual dimorphism in that males develop diabetes earlier and much more severely than their female siblings. Most male IRS2 knockouts die by 12 weeks of age due to complications of diabetes. These results have been published now for many years. And this is our basic model that we work with in the laboratory. Luke Noon, Ramon y Cajal, a researcher, very talented, very bright guy. Also developed another model in the laboratory, as well as a series of tools to modulate hepatic differentiation. These HEPA-RG cells were developed by a group of hepatologists in Rennes in France. HEPA-RGs are bipotent hepatoma-derived cell line, which can be differentiated under very defined conditions to generate these islands of hepatocyte-like cells, which display both morphological as well as functional characteristics of hepatocytes. Luke developed a series of tools, which you're going to see in the next few slides, to monitor this hepatocyte differentiation in this HEPA-RG model in order to define in which stages IRS2 signaling intervenes to either promote or impair this process. So you'll see these different markers of hepatocyte differentiation in the next few slides. One of the basic conclusions that we derived from these early studies, and these experiments are related, this whole study is related to our participation in two FPF7 projects, both of which had the goal of developing progenitor cell models, embryonic stem cell models for cell replacement therapy in chronic liver diseases. And one of the basic observations was that insulin is required for the differentiation of this progenitor model. This was one of the first observations that we had, the first hints that insulin signaling was such an important modulator of the differentiation of these liver progenitors. By the way, Javier, this is an in-cell analyzer, and this is the sort of data that one can generate. These outlines are actually what the machine uses to count and quantify these cells. Very useful. So that's one of the fundamental conclusions. And in the next few slides, the studies also show that IRS2 is the principal intracellular mediator of the effects of the the positive effects of insulin on the differentiation of these liver progenitor cells to functional hepatocytes. The profile of IRS2 expression as well as IRS1 is very interesting, such that during the early phases of differentiation, the levels are high. As the cells proceed through the differentiation process, the levels of both these insulin signaling molecules are downregulated. And this has important consequences for the appropriate regulation of the expression of these molecules. These experiments in which we have silenced IRS2 or overexpressed IRS2 have enabled us to conclude that without insulin and IRS2 signaling, the HEPA-RG cell line does not differentiate efficiently to hepatocytes. And when we do the converse experiment to overexpress IRS2 in these progenitor cells, the jazzed up expression of IRS2, the increased sensibility to insulin actually drives the hepatocyte differentiation. These observations in this HEPA-RG model provided a basis for continuing these studies in what is perhaps a more relevant physiological model, which is using hepatic damage through the administration of a DDC diet to test the role of IRS2 in vivo. This summarizes some of the data which I've just shown you, whereby insulin signaling promotes this differentiation process. And our studies demonstrate stress, I would say, that early IRS expression is needed to trigger, to drive the differentiation process. And subsequently, these insulin signaling modulators need to be downregulated so that the hepatocytes mature to acquire CYP function, to acquire lipogenic function. When we've manipulated IRS2 expression between these two phases, we have observed defects in different capacities of these hepatocytes, both to respond to damage as well as to respond with an appropriate sensitivity to insulin. As I mentioned, these very basic studies have enabled us to conclude that IRS2 is important in progenitor cells and has allowed the basis for testing how this molecule may regulate this process in an in vivo setting. Fatima Manzana, a predoctoral student in the lab, who's about to finish her stay in our laboratory. She's at the point now when she has to think seriously about sitting down and writing up all the results I'm going to show you today. This has been one of the aspects of her doctoral thesis, to assess the role of IRS2 in the response of the liver to this DDC diet. Why did we choose the DDC diet rather than another form of liver injury or insult? And the basic reason is because we were interested in analyzing the role of progenitor cells in the ductal response, and therefore this particular method was more consistent with the particular aims of the study. As you can observe in panel B here, the treatment with this diet in wild-type mice induced an upregulation of IRS2, consistent with our working hypothesis that IRS2 signaling may be involved in progenitor cell response and in mounting an effective repair mechanism for responding to damage. The IRS2 expression declines with the treatment with DDC. Consistent with the upregulation of IRS2 expression and perhaps a peak of damage at day 7 of this treatment protocol, you observe upregulation, increased activity of liver damage readouts, ALT and AST. It's very interesting that both of these markers of liver damage decreased at day 14 in the wild-type animals, whereas in the IRS2 knockouts, both of these indicators of liver damage remained elevated, suggesting that the absence of IRS2 may disable the mechanisms for responding to this particular protocol for inducing liver damage. Based on these observations, we then wanted to look more specifically at the process of what would underlie a typical scenario of responding to liver damage, in which hepatic stellate cells may be activated, and the remodeling of epithelial components, which underlie much of the pathogenesis related to fibrosis and other aspects of the progression to NASH once there's an accumulation of lipid deposits within the liver. Around this time, actually, we had invited the speaker. This publication appeared after we had developed the HEPA-RG model, demonstrating that FGF7 is a very important molecule in regulating the niche signaling and the process of responding to liver injury. The FGF7 knockout mice are incapable of mounting a sufficient response to liver damage, and for other factors also display a reduced survival rate. As many of you know, because it's a privilege to discuss these findings with a very knowledgeable audience, that during hepatic injury, hepatic progenitor cells proliferate and form duct-like structures in periportal tracts. And these liver progenitor cells are hypothesized to be surrounded by stromal cells which produce the molecule FGF7. And in a very elegant feedback mechanism, the liver progenitor cells are predicted through this model to express the receptor for this molecule, FGF7. Sorry, but the menu button is right next to the one for the pointer. So this feedback mechanism whereby FGF7 may be secreted by hepatic stellate cells, and the receptor for this growth differentiation protective molecule is on the liver progenitor cells, provides a very interesting model. I'd also like to comment to you that the receptor for FGF7 is the FGFR spliced form, and we'll get to that information in just a moment as we go through the steps to actually test how IRS2 signaling may interact with the FGF pathway to regulate the response to the DDC diet. In the continuation of these studies, I'd also like to mention that Maria Jose Arembol, another pre-doctoral student in the lab, has made significant contributions, as the objective of her thesis is precisely to define these crosstalk mechanisms between FGF7 and the IRS2 pathway in regulating the response of liver progenitor cells to injury. I'll get back to this model. This is a summary. I'm giving you a view of the results and of the model, interactive model that we've developed from the results. And now I'll explain a little bit about how we arrived to these conclusions regarding this, this feedback model. So using the IRS2 knockout model and age-matched wild-type controls, as you saw in the previous experiments, we looked at the expression of a series of markers for hepatic function. We observed this very interesting, and this is the basis of the working hypothesis to look at the crosstalk between these two pathways in the liver. FGF expression was clearly upregulated in response to DDC diet at day 14 and day 21 in the wild-type mice. However, in the IRS2 deficient liver, this molecule was not as significantly upregulated, and you also see an impaired, a dampened response in terms of EpCAM as well as osteopontin, SPP1 positive cells within the livers that have been treated with the DDC diet. This then led us to characterize in more detail these distinct cell populations that are related to a progenitor cell response in terms of effectively activating liver repair. And again, we see that in wild-type animals, GFAP positive cells were maintained throughout this diet treatment, whereas in the IRS2 knockouts, we see a significant dampening of the effects of the treatment on GFA positive populations. For the next series of experiments, we went back to in vitro models to test the relationship between FGF signaling and IRS2 expression and function. And in these particular series of experiments, you can see, again, in the treated livers, in the wild type, in the IRS2 knockout mice, the incidence, the detection of cells that are undergoing apoptosis is significantly elevated in comparison to a wild type liver. These measurements were made at day 7, which is precisely, I just remind you about the earlier data, this is precisely the point in the treatment protocol when we had observed an upregulation of IRS2 expression. And these results suggest that the absence of IRS2 signaling promote apoptosis and may, this lack of a protective effect may underlie the exaggerated response to the damage in the IRS2 liver. One of the basic proposals that we have is that IRS2 may be regulating the expression or secretion or binding of FGF7 in this process of activation of the hepatic stellate cells in response to damage. We've used a human cell line of hepatic stellate cells. And Fatima and Maria Jose have developed a very elegant co-culture model. I'm not going to show much of that data today. And in these experiments, which reinforce, which confirm what we observed in vivo, if IRS2 is silenced in the hepatic stellate cell line, we observe decreased expression of IRS2 and an increased susceptibility to apoptosis. So these in vitro experiments nicely support the observations that we made in vivo using treatment with DDC in our IRS2 mouse model. We tested this relationship, this loss of FGF7 function in the IRS2 knockout mice, again in our in vitro model of the HEPA-RG cells. We treated the cells, also we treated mice, which is shown here, and I think these data are much more relevant when we treat the IRS2 deficient mice. So restoring the FGF7 levels and using as a readout the activation of the ERK pathway. In wild-type animals, you see an activation based on the presence of phospho-ERK in the treated animals, whereas in the IRS2 knockout mice, the response to FGF7 is blunted. This led us to the next few experiments, which test whether this defect is simply limited to the production, to the expression and secretion of FGF, or whether it might also be related to the receptor for FGF7. As it turns out, it may be both of these things. And I'm going to go now rather quickly because I think we're getting close to the end of the time. And given that tomorrow's holiday, I certainly don't want to delay you. So again, we have tested this hypothesis using the HEPA-RG line. When we apply recombinant FGF7 to the HEPA-RG line, the effect is similar to insulin. in that it promotes hepatocyte differentiation. You can see also from the enhanced expression of the marker ApoA2 that these effects are very similar to insulin. What we had observed in the cell lines, so we've used cell lines. Luke has developed stable HEPA-RG cell lines where we've silenced different components of the insulin signaling pathway from the receptor to IRS1, IRS2, and other components of the signaling pathway. And when IRS2 is silenced in these cell lines, we observed, in contrast to the in vivo results, that FGFR expression was not altered, the total FGFR, but rather the defect related to the loss of IRS2 expression is more specific for this alternatively spliced form of the FGF7 receptor. So the FGF receptor 2, this 3B spliced form. This sets the stage for a very interesting feedback mechanism whereby IRS2 may be modulating both the expression of the factor FGF7 as well as its receptor. Recall that the molecule and its receptor expressed in different compartments of liver, such that the progenitors are likely the ones to be expressing this form of the spliced receptor, whereas the hepatic stellate cells may actually be the source of FGF7. Yeah, this I think is just what I was commenting. What I would encourage Rafa and Jordi to do is, I realize your calendar is full for the next year, is to invite Luke to come next year and provide details related to this mesenchymal epithelial transition and some of the more detailed work he's done in defining epithelial remodeling using the models that we have available in the laboratory. This is just a summary of what I've commented about the source of FGF7 expression and secretion and the expression of the FGFR3B receptor in liver progenitor cells. These are a series of other experiments which we've done in the HEPA-RG cells to further confirm that these effects are specific to the 3B receptor. I'll try to finish up in a couple of moments. Based on the results that have accumulated in the lab during the last 6-8 years, we propose the model, which is represented here. And all of these beautiful models and schematics are the work of Luke, who's not only a great scientist, but he's also a good artist and very brilliant in conceiving these sorts of visual models. So during the differentiation in our model, HEPA-RG cells, IRS2 expression is upregulated in the early phases of differentiation and needs to be downregulated for the maturation process in the later stages of hepatocyte differentiation. In the new data that I presented today, this expression of IRS2 parallels the expression of the alternatively spliced form of the FGF7 receptor, the FGFR2/3B receptor. You can see a similar profile during the differentiation of this in vitro model of hepatic progenitor function. This is another piece of evidence that we believe links this the function of IRS2 signaling, both in the hepatocyte differentiation program as well as in the response of liver to damage. So Luke and the two students in the lab, Fatima and Maria Jose, have this work under review. It's the second revision, and I hope when Luke comes to speak in the next series of these seminars, the paper will be published by then. But they nicely describe it. These data suggest feed forward, so a positive feedback loop whereby IRS2 during a certain phase of differentiation, activation of progenitor cells may regulate the expression of the certain forms of the FGF receptor. And this, we have data from the in vivo studies to confirm that this also happens in response to liver insult, such as the DDC diet. In parallel, IRS2, as I've shown you today from the data, IRS2 signaling may also be very important in regulating the survival of cells in the stroma, in preventing apoptosis, so that FGF7 is secreted in appropriate amounts and with the proper timing during the differentiation activation response in order to properly repair damaged areas or during other aspects of liver function. Luke has continued this story in a much more detailed way, and I think it would be a very interesting part 2 of this story for next year about the details about the IRS2 mediated regulation of stroma epithelial interaction and how insulin is required through IRS2 to co-regulate this process with FGF7. These findings are novel and suggest a new pathway whereby interventions could be developed both to prevent damage as well as potentially to reverse damage in a hepatic context. The data that we have in the lab, and these are not in the talk, it's simply an anecdotal comment, both in vitro and in vivo, we observe that if we can restore IRS2 expression, we're able to revert pathological processes such as accumulation of lipids in the liver. Presumably, this is through the restoration of sensitivity to insulin, and perhaps now that we've defined this new mechanism, through the restoration of physiological crosstalk with the FGF pathway. And just in the last minute, I just want to, this is to define the inflammatory components of this crosstalk pathway and how IRS2 responds to inflammatory components involved in liver damage is also an ongoing study in the lab. But in the next slide, I just wanted to leave you with a message about pancreatic beta cell function. And I couldn't give a talk without mentioning the interaction between the pancreatic beta cell and the liver in the development as well as progression of metabolic diseases. As you know, the liver produces a series of growth factors as well as inflammatory cytokines. In insulin-resistant states, glucose production is elevated, and this puts additional stress on the pancreatic beta cells to respond to an increasing demand with enhanced insulin secretion during an adaptive phase. As long as the beta cells are able to keep up with this increased demand, for example, in a chronic hyperglycemic state, metabolism is maintained more or less in a normal range. However, when the beta cell mass can no longer compensate for insulin resistance, particularly at the level of the liver, which the liver is potentiates the whole panorama of metabolism related to hyperglycemia. So when the beta cell mass declines due to increased the pernicious effects of lipotoxicity, as well as hyperglycemia, diabetes obviously develops and progresses. Today I was discussing with Rafa's group about the expansion of the liver mass and the profile in the spectrum of liver disease. And in the same way that you may have an increase in liver mass in the early stages of steatosis, for example, clinical and basic studies also show that beta cell mass expands in response to insulin resistance. And our laboratory is very interested in defining what are the molecular mechanisms underlying this beta cell compensation and why they fail and diabetes ensues. And I think this perspective of the beta cells is highly relevant for the increase in, for example, liver volume and other components that are related to the vicious circle that is reflected by steatosis and the progression to fibrosis. So not only should we be looking for targets to modulate steatosis and fibrosis, but also to modulate, to enhance, to extend, to prevent the failure of this beta cell compensation, because it's also another level, another strategy for preventing the progression of the pathological scenario that leads to cirrhosis and in a percentage of the cases to hepatic carcinoma. The relationship between metabolism and tumor development is also studies for another day, a story for another day. I thank you all very much for your attention, and I'd be happy to take your questions. If you could just go to the final slide so I can acknowledge. This is a new model which we've developed, which allows us now to track, in the next slide, IRS2 expression. Actually, this has been a limitation throughout the years because the antibodies that we have don't work very well for histological sections for localizing the temporal spatial expression of IRS2. And we've recently developed this green mouse, an IRS2 GFP reporter mouse, which will now allow us to perform more detailed, perhaps more interesting studies, both in the liver as well as in the pancreas, to understand what factors, what physiological, what pathological factors, how diet, how different components of different factors may influence IRS2 function and expression, with the ultimate goal of trying to develop drugs or lifestyle interventions that would preserve IRS2 function in both the liver and the pancreas as a means for avoiding long-term chronic metabolic disease. And with the last slide, I just want to acknowledge to my lab. This is my lab plus our neighbors. This photo was taken recently for World Diabetes Day, the 14th of November. I've already shown Luke, who's the principal driver of the projects that I've shown today, Fatima Maria Jose, and also a new postdoc in the lab, Esperanza, whose project is to use the new IRS2 GFP model to further characterize the role of IRS2 in the pancreatic beta cells. We're also grateful to our funding agencies. Many thanks to the Ministerio and Carlos Tercero for funding Crazy Ideas during almost 20 years now that I'm in Spain. Crazy innovative. We're very grateful for their support. I'm a member of the FIBERDEM. I have several investigators in my lab that are financed by the FIBERDEM. And this work started, as I mentioned before, through FPF7 project in Overlive. The Centro de Investigación Príncipe Felipe, we have an open call for recruiting young as well as senior investigators. We're particularly interested in recruiting new groups to strengthen the programs in cancer as well as in metabolic diseases. Thank you very much. I'll be happy to answer your questions. We're also very interested in that question, and I think it has a lot of clinical relevance. This is precisely one of the questions that we'd like to address with the new GFP IRS2 model, because I think these sorts of questions can be better addressed in vivo than in vitro. However, I'll try to answer it a little bit with a HEPA-RG model, this cell line which we use as a model of progenitor cells. We can show by modulating IRS2 expression, so the duration of IRS2 expression, some of the plasticity that these immature progenitor cells have to differentiate to the different lineages. I would say the preliminary observations that we have so far is that IRS2 expression definitely marks, I wouldn't say determines the fate, because I think that's a much more definitive, a term much more related to developmental processes, but it certainly favors the differentiation to hepatocytes because the IRS2 pathways ultimately will regulate aspects of hepatocyte function that may not necessarily be required for biliary cell function, right? I think this is a very interesting component of what I mentioned as plasticity before, in terms of trying to modulate different cell phenotypes within the liver environment. I would, in a way, compare your question a bit to also what we're very interested in the pancreas. It's the same concept. So can we reprogram? Can we convert an exocrine cell or an alpha cell to a cell that produces insulin? The answer about IRS2 in that context is much clearer. IRS2 expression is specific for a beta cell phenotype. Based on the analysis that we've done so far, it can be involved in the reprogramming of other endocrine cell types to a beta cell phenotype. In the liver, and as I mentioned, these are preliminary observations, but it would suggest that IRS2 may be more specific for hepatocytes as opposed to other cell types. It definitely could be. The concept of very localized IRS2 signaling is also one of the models which we're testing. So rather than the whole liver being sensitive to insulin, actually looking at different zones and how local insulin signaling may be regulated by that. The basis of your question, we could easily test in vivo in the IRS2 knockout model, for example, to see whether they have an impaired or enhanced ductal reaction. The problem is that these animals don't live long enough for us to actually study that process in the whole animal. As I mentioned before, these animals develop hyperglycemia very rapidly. And given that a ductal reaction would be an accumulative process or over a period of time, these animals die around 8 to 10 weeks of age which really limits them as a tool for addressing this question in vivo. Certainly, some of the results I've shown today would suggest it may be more related to the process of fibrosis, for example. And you've mentioned that there's heterogeneity, there's variability in patients that present with NAFL. This variability may also be related to the degree of insulin resistance which they have, because the tools that we have for assessing the degree of insulin resistance in these patients is still, quite frankly, not very sophisticated. I think probably all of these patients, irregardless of the degree of insulin resistance, are going to have some degree of lipid accumulation, right, in the hepatocytes. And certainly IRS2 is involved in this abnormal accumulation of lipids, either through a decrease of IRS2 function or conversely through a gain of function, because we observe that in both cases. The clinical studies thus far, which have looked at the role of IRS2 in patients, have not been very conclusive. The studies have been oriented towards a genomic level, a genetic level, looking at IRS2 mutations, for example. Our observations from the mouse model, from cellular models, would suggest that it may be much more a question of the level of the expression of IRS2 protein expression. And therefore, one of the determinant factors would be in these patients, what is the average half-life of the IRS2 protein in the liver of these patients. And we know that different inflammatory cytokines alter the half-life of IRS2 by stimulating degradation or altering the subcellular distribution of it. So there may be also variability of IRS2 expression related with the heterogeneity of the disease process.
So we are going to close now.
Thank you very much.
Thank you very much, Deborah, for coming here.
Have a nice long weekend to those of you who have Puente.
Bye-bye. Thank you.